Self-assembly in nanobiotechnology is a spontaneous, self-ordering, and reversible organization of structural components, monomers, into more complex nanostructures. So this is the cumulative effect of many molecular interactions between functional groups and is found in many important biopolymers, for example, the proteins and also nucleic acids, and give them specific functions. Now, for example, the nucleic acid, as we all know, are polynucleotides that consist of different types of nitrogenous bases. These include adenine, guanine, cytosine, and thymine or uracil. These combinations, there are a lot of combinations possible, and these unique combinations allow them to encode genetic information while also allow them to form into specific structures with complex functions. Now, how does this work? The self-assembly in nucleic acid primarily depends on two forces. One of them is base pairing and the other one is a base stacking. As you all know, the base pairs pair between complementary bases. For example, adenine and thymine form 2 hydrogen bonds between them, while cytosine and guanine form 3 hydrogen bonds. This formation of hydrogen bond between the complementary groups, the polar part of the complementary groups, give them specificity, while this effect is further stabilized by the formation of hydrophobic interactions between adjacent base pairs. The formation of base stacking effect between the pi electrons resonate with one another to stabilize the entire double helix structure. And both of these give them the stability to self-assemble. However, it's the base pairing that gives them specificity and allow us to manipulate, which we will go into later. Now, for example, take this tip. Imagine this individual teeth are the base pairs, and this unique sequence is known as the primary structure of nucleic acid. With a complementary base pair, we can make it to hybridize into a stable duplex, and this is known as a secondary structure. The double helix we are so familiar with is part of this secondary structure. However, this gives them high stability as a storage of genetic information. But that's not all. There's a lot more we can do with this. For example, with palindromic sequence, we can let them self-hybridize and form a lot of weird structures. They can also only partially hybridize to form half duplex, while some of the single strands are exposed. This gives them a lot of different functions. For example, we know that tRNA is in a clover shape. The specific sequence allows them such that only parts of them are hybridized, and they can bind with different other components, for example, the amino acids, while the other parts can also recognize and identify the specific codons to which they bind to in the ribosomal mRNA complex. Not only that, they can also fall into tertiary structures where complex tertiary interactions between the helix chains. So this can be found in several different RNA with catalytic functions known as a lysozyme. They can even further form into quaternary structures. For example, DNA with histone form the chromatin, and rRNA and proteins form the ribosome. So these are all made possible because of the unique nucleotide sequences. With the appropriate sequence, you can make it do a lot of different functions. And this is what the nanobiotechnology is trying to do. We want to manipulate it and to synthesize specific sequences that allow it to do different stuff, all kinds of different functions.
In cell, DNA cell assembly is done through the help of assembly factor like binding protein, but in lab, we can also manipulate it, like for example, DNA origami. So DNA origami is a technique that allows the precise assembly of DNA origami object in nearly any desired form with the assistance of computer programs. By utilizing DNA origami template, the structure can be precisely addressed. The site-specific modification of fundamental fragment can be made. This modification, along with the biocompatibility and the biostability of DNA origami nanostructure, make them suitable for targeted delivery and control release of therapeutic cargoes. So below there's three different DNA origami, and it's three different shape. So this shape is being folded and become a different shape, and the DNA origami can be used to fold it and become a specific shape for the specific function in the DNA label. Yeah. And then next, in the biomedical field, this nanobiotechnology has many applications. For example, first of all, is the improve of the drug delivery. So aptamer is one of the nucleic acid that being used for the improvement of the drug delivery. So aptamer is the small oligonucleotide sequence which can be used for targeting in drug delivery system. So it is highly versatile and allowing for efficient and rapid identification of the target, such as small organic and inorganic molecule, protein, cell, and even tissue. It can also mediate the entry of nanosystem into cancerous tissue or cells. Next, we talk about we can the DNA cell assembly can also use to improve the drug stability. So the instability of the single-stranded nucleotide drug such as the siRNA and the enzyme often limit their use when administered alone. So to overcome this issue, Go et al. Utilized the hybridization property of siRNA and aptamer with DNA to bind them to tetrahedral DNA nanoparticle, or we can call it as TDN, which are classical three-dimensional structures that possess high stability due to their 6 edges and 4 vertices. The inclusion of a folate-mediated system in the structure target folate receptor upon cell entry, enabling the release of aptamer with siRNA. This results in gene silencing, promoting intratumoral infiltration of cytotic T cell. And then the last application in the biomedical field is the drug control release. So to control the release of a drug at a target site and at a specific time, the assembly and disassembly of the DNA strand need to be precise control, means that it's very important. And then control release of these nanoparticles is depend on a specific internal and external stimulus trigger like temperature, acidity, light, and ultrasound. And then below there's one of the example that has been put that's related to the temperature that control the release of the nanocarriers. So in 2020, Zhou et al. Introduced a DNA nanocage that exhibit a temperature sensitive response for intelligent encapsulation and release of enzyme protein, which is Horseradis peroxidase, which in the short form is HRP. So in their study, they show that at 37 Celsius, the double helix level of the DNA nanocase decreased, resulting in a more flexible structure that allowed the HRP to enter. And then when the temperature was low to 4 degrees Celsius, the double helix and hydrogen bond structure will restore and encapsulating the HRP. And below there we can see that at first at the 4 degrees Celsius, there's nothing inside. And then when the temperature increased until the 37, the double helix level of the DNA nanocase will decrease, and the HRP can be enter it. And then we lower down the temperature to the 4 degrees Celsius, and the double helix and hydrogen bond structure will restore and encapsulating the HRP. And then when it comes to the target site, and then we will increase back the temperature so that the HRP will be released. So in conclusion, the nucleic acid cell assembly study has high potential to be applied in the nanobiotechnology and biomedical field. But there's also a challenge remain for this technology. So particularly, we need to improve the long-term stability and cost effective for the large-scale production in this technology.